Hi everyone, my name is Sarah, and in this video we're going to be discussing the respiratory gas exchange and specifically the role of hemoglobin. So shown over there to the right on this first animated picture, you can see that we're going to be discussing how the oxygen that we take in our lungs travels to the heart before being dispersed out into the tissues, and how this process is facilitated by the binding to hemoglobin. So shown here in picture A is a red blood cell, and picture B, that blood cell has been blown up to show what is inside, specifically that hemoglobin molecule containing the oxygen bound to its heme group, and that's going to be a central role in this video. So a little bit of a background to begin. What exactly is hemoglobin? Hemoglobin kind of has a dual role. It is contained in red blood cells, and it both efficiently carries oxygen from the lungs to the tissues of the body, and then it's also going to help in the transportation of carbon dioxide and hydrogen ions back to the lungs. And hemoglobin is a protein, again, found in red blood cells, and if you look at that picture there on the left, it's comprised of 2 alpha and 2 beta subunits that surround an iron-containing heme group. Each subunit surrounding the central heme group is going to contain iron and bind 1 oxygen molecule. So then in total, that hemoglobin molecule will be able to bind 4 oxygen molecules. Oxygen can bind readily to this heme group, and the ability of oxygen to bind increases as more oxygen molecules are bound to the heme. And it's also important to know that there are several disease states and altered conditions, such as a few genetic disorders, that can affect this binding ability of oxygen and either increase or decrease its ability to dissociate from hemoglobin, which is going to have a number of detrimental effects. That's just something to kind of note the impact that that can have. So oxygen binding is going to induce substantial structural changes at the iron sites in hemoglobin. Again, oxygen binds to the heme component of the protein hemoglobin in the red blood cells, and this process occurs in the pulmonary capillaries adjacent to the alveoli of the lungs. And the oxygen molecule is going to bind at the 6th coordination site on the iron ion, and this occurs because the other 5 sites are already taken or already filled by nitrogen, and so the 6th coordination site is the only one that's open. And this binding is going to substantially rearrange the electrons within the iron ion, which makes that ion become effectively smaller, and it's going to allow it to move better into the plane of the porphyrin. And on the oxygen binding, as shown here in that picture to the right, there's going to be substantial changes in the quaternary structure that corresponds to this T to R state transition that is occurring. So again, over there on that picture, we have the T form shown of hemoglobin before it goes through this conformational change to the R form of hemoglobin, and you can see that the initial T form was more deoxygenated and it had that low oxygen affinity, but once it binds and goes to the R form, it has that higher oxygen affinity, as the oxygen binding is going to disrupt those interdimer bonds. So then what does the binding capacity of oxygen to the heme look like in the lungs versus the capillaries? There's going to be a few factors that are going to impact the binding capacity of hemoglobin. A few of those are going to be the partial pressure of gases, temperature, and pH. And I like this graph shown to the right. I think it does a good job of showing the different factors and what that shift is going to look like on the oxygen hemoglobin dissociation curve. So as shown there, the high pressure of oxygen, the low temperature, and the low pH are going to aid the binding of oxygen to hemoglobin in the lungs. The opposite then is going to be true for the capillaries and the tissues. Oxygen must be transported in the blood from the lungs because the partial pressure of oxygen is going to be relatively higher, approximately 100 torrs, than in the capillaries and the tissues, where there the partial pressure of oxygen is much lower at approximately 20 torrs. Another way to look at that, in the lungs, the hemoglobin is going to be saturated with oxygen at approximately 98% of all oxygen binding sites occupied, whereas when they reach the capillaries and the tissues, that saturation level drops to 32%, which is why the oxygen is getting dispersed out into the tissues. So on kind of a side note here, how will carbon dioxide interact with water and affect the pH of capillaries? Well, CO2 is acidic in nature, so when it interacts with water, then the pH of capillaries decreases. A decrease in pH is going to shift that standard curve towards the right, as shown there in this graph, the low blood pH and the high carbon dioxide. And this occurs because at greater hydrogen ion concentrations, various amino acid residues, such as the histidine, are going to stabilize the deoxyhemoglobin in the T state, and that T state has a lower affinity for oxygen than the R state. And so with the increased acidity, the hemoglobin is going to bind less oxygen for the given PO2, and that's going to be more of the hydrogen ion. So overall, this is known as the Bohr effect. And so the Bohr effect is the physiological phenomenon that describes the decrease in the amount of oxygen associated with hemoglobin and other respiratory compounds in response to a lowered blood pH, resulting from, again, the increased concentration of carbon dioxide in the blood. So thanks for watching. I hope you learned a little bit more on the process of gas exchange, specifically the way the oxygen binds to hemoglobin and a few of the factors that affect that, as well as the role that carbon dioxide and pH can play.